All right, I think we can get started. Hello, everyone, and welcome to another edition of Science Cafe with the Conservation and Research Department at the Atlanta Botanical Garden. We're really happy to have with us this evening Dr. Elizabeth Hermsen, who will be presenting on the diversity of the Neogene gray fossil site paleoflora and its importance to understanding the modern flora of Eastern North America. Dr. Hermsen is, I didn't mention, but she is a research scientist with the Paleontological Research Institution in Ithaca, New York. Before we get started, I just want to go over a few things and let everybody know that we are recording this. So if there are people who are unable to attend that you think might be interested, this will be available on our website for folks to listen to at a later date. And I will share that link at the end of the presentation. Additionally, we encourage folks to ask questions, and we will have a question and answer section at the end of the talk. If you do have questions, if you look at the bottom of your screen, you should see a little feature that says Q&A. If you could direct any questions you have there, as opposed to in the chat, and then at the end of the talk, we'll try to get as many of those questions answered as we possibly can. The only other thing I'd like to do is just give you all a little bit of background on Dr. Hermsen and what she's been up to of late and with her career. So Dr. Elizabeth Hermsen received her BS in botany and geology from the University of Wisconsin-Madison and her Ph.D. from Cornell University. She held postdoctoral positions at the University of Kansas studying fossil plants from Antarctica and Cornell University studying fossil plants from Patagonia, Argentina. She later worked at Ohio University in Athens, Ohio as a faculty member. Today, she is a research scientist at the Paleontological Research Institution in Ithaca, New York. In addition to research, she helps edit PRI's Journal Bulletins of American Paleontology. She also contributes to the development of PRI's Daring to Dig, Women in American Paleontology temporary exhibit and companion website, and is writing the section on fossil plants for the Digital Encyclopedia of Ancient Life. So I think without further ado, Dr. Hermsen, I'm ready to give you the floor.
All right, thank you. So let's see if I can. Okay. There we go.
All right, that's good.
Great. Hello, everyone, and thank you for inviting me here today to give this talk. I'm really excited to share my research with you and share just the great fossils that come out of Gray Fossil Site. So before I start talking about the site and my research, see if I can advance a slide. There we go. I would like to do my acknowledgements just so they don't get lost at the end. So a lot of people have supported me and helped me in this project, and I'd just like to acknowledge a few of them. For the Cavalignum project, which is one of the projects I'll be talking about, that was co-authored with a student, Carolyn Siegert, who now happens to be a graduate student at Cornell, and also another student helped with the project, Stacy Welker. The Coralopsis project was a project I did with Zach Quirk, who is now at the University of Michigan pursuing his Ph.D. Funding for this research came from Ohio University and also the National Science Foundation. I'd like to thank Maria Gandolfo for loaning me use of her stereo microscope, which allows me to document the flora. And last but not least, the Gray Fossil Site curators and staff, students, interns, and volunteers. They've hosted me a few times at the site to work in the collections, and they also do all of the fossil cleaning and sorting and a lot of the curation work. So they deserve a big thanks for making this project possible. So what is Gray Fossil Site? Where is it? Gray Fossil Site is a site, fossil site in northeastern Tennessee. So I don't know if I have a, it's in the northeastern part of Tennessee, right where the spade is on this map. It's near the city of Gray. There is a little larger city nearby called Johnson City, if you're familiar with the area. Down in the lower left-hand part of the screen, you can see a picture of the Gray Fossil Site Museum. So there is a museum that's built right on the site, not of course where the fossils are, but sort of in front of the main excavation area. And the fossils are excavated kind of from behind the museum. So there's a field season every year when excavation takes place. Up in the upper right-hand side of the portion of the slide, you can see the outdoor portion of the site. So there are a couple of shelters where part of the fossil preparation takes place. And there's also an area of excavation off to the right. And on the bottom right-hand side of the slide, you can see one of the displays in the museum. So there is a nice little museum on the site where some of the vertebrate fauna has been reconstructed and is on display. So how old is this site, and how do we know how old it is? Well, the site is considered to be Neogene in age. So Neogene is a time division known as a period, and it goes from about 23 million years ago to 2.6 million years ago, and it's divided into two epochs. And those epochs are the Miocene, which is about 23 to 5.3 million years ago, and the Pliocene, which is about 5.3 to 2.6 million years ago. Gray fossil site itself is currently thought to be early Pliocene in age. So the age estimate is about 4.5 to 4.9 million years old. Where does this age come from? Well, it comes from analysis of the fossils at the site. So basically this chart down here in the lower left-hand side of the screen shows the age ranges of various fossil animals that occur at the site. And these ranges, of course, are taken from other fossil sites where they're found. And the overlap in these ranges gives an approximate interval of dates that the gray fossil site might conform to. So that's where the age comes from. Gray fossil site is really important because we don't have a lot of plant fossil sites from the Neogene of Eastern North America or the Eastern United States. So the map here shows the Neogene fossil sites in Eastern North America that have produced a fair number of macrofossils. And they're scattered all over the Eastern United States, and there aren't that many of them given the time span involved, about 20 million years. So there are a few Miocene sites. Brandon Lignite, which is in Vermont, is the oldest of these, and probably also one of the more diverse or interesting. And then there's Brandywine in Maryland, so that's late Miocene, and two middle Miocene localities, Allen Bluff, which is in Florida, and Hattiesburg, which is in Mississippi. So in the Pliocene, there are even fewer sites. Gray fossil site, which is in Appalachia, and Pipe Creek Sinkhole, which is in Indiana, are near in age. They're both early Pliocene. And then Citronelle is down in Alabama. So really the richest Neogene record is along the Gulf Coast. There are a lot of little sites that haven't been well explored, and there are many fewer sites outside of that area. So why does gray fossil site occur where it's occurring, or why aren't there other sites around? Well, there's a good reason for that. And the reason is that the bedrock in the area where gray fossil site occurs is actually quite ancient. So it's an ancient carbonate bedrock, sort of like similar to a limestone, and a sinkhole formed in this bedrock in the Neogene. So basically water moving through the bedrock starts to dissolve it, and it forms a cavity. And if the roof of this cavity becomes too weak, it can collapse and form a depression. So in the lower left-hand side of the screen, you can see one of these depressions in the modern day. Sometimes sinkholes open up in places like Florida because they have a similar geology. So this can happen. So a depression forms. That depression can fill with water and form a lake or pond. So it's thought that gray fossil site is actually an ancient sinkhole, lake, or pond. And the fossil deposit is actually preserved in the lake or pond sediments. So around this ancient a water hole, essentially, there was a forest. That forest was dominated by oak, hickory, and pine. And we know that because people have done the palynology of the site. So they've looked at the pollen and spores that occur in the sediment. And these are some of the more common taxa. There's some debate about how dense this forest was around the site. So another thing we might wonder is, what was the climate at this time? Earth's climate has changed over time. Was Gray Fossil Site's climate like it was today, or was it different? A number of studies have been done looking at this, and they've looked both at the plants, especially the pollen, although I think there was also one done on leaves more recently, and then also at isotopes. And so the conclusions of, basically the general conclusions from these studies, is that Gray Fossil Site was probably similar in terms of how much precipitation or rainfall it was getting in the past, but warmer in the past than it is today. And in particular, there was probably less variation from season to season and what the temperature was like. And importantly, winters were probably warmer. So there are some indicators of this. For instance, alligators are known from the site, including young alligators, which are not particularly cold tolerant. There are also beaded lizards. So these are things related to, for instance, the Gila monster of the southwestern United States. So the most recent study on the climate at Gray Fossil Site did some useful comparisons to modern climate. And the authors concluded that the mean annual temperature at Gray Fossil Site was like that of Atlanta, Georgia. And the rainfall was probably similar to Tampa, Florida. So basically, the climate at Gray Fossil Site would be as if you had moved lower in latitude in the United States today. Gray Fossil Site is probably best known for its fauna. It has a very diverse vertebrate fauna. There are mammals, there are reptiles, there are amphibians, I think there are some birds. So there's a whole bunch of material that's come out of this site that's really exciting in terms of the animals. And this slide just shows some of them. The site's really well known for producing a lot of tapirs. So there are many tapirs that come out of the site. In the lower right-hand corner is the skull of an extinct North American rhino. There are also red pandas that come out of the site. This is probably one of the more well-known fossils from the site because this red panda occurs today in Asia, but is extinct from North America. And another kind of interesting megafaunal member from the site is the mastodon. So mastodons do come out of the site, and there's a picture of a jaw here and also some tusks. So what about the plants? I'm working on the plants, and the plant, the flora is very rich, but the fossils aren't maybe what we think of normally as plant fossils. So on the left are maybe some typical plant fossils. If you just, search plant fossil in Google Images, things like this might pop up. The top left here, we have some leaf imprints. So leaf impressions or compressions, if they've got a little carbonaceous film on them, are very common plant fossils. Things like petrified wood shown on the lower left-hand side of the screen. And also, since I'm in New York, here in the center, there's a nice stump from the famous Gilboa flora, which is our forest, which is one of the oldest known forests in the world. Gray Fossil Site plants, however, don't really look like these fossils. So the Gray Fossil Site plants are shown on the right. And at the top right here is a display of these Gray Fossil Site plants. And yes, there are fossils in this display case. They're much tinier than the labels next to them. So that's the first thing to notice about the fossils in this flora. They tend to be very small. On the bottom right here is a Petri dish. This is actually on a microscope that shows just a selection of plant fossils. This would have been something I was sorting when I visited Gray Fossil Site. So again, very small. Many of the fossils at the site are probably about 1 centimeter or less in total length. There are some things that are bigger, but a lot of things are small. And then on the bottom right here is an image taken with a microscope of a fossil. The scale bar in the corner of that image is 2 millimeters. So that particular fossil is maybe a little less than a centimeter in length. So these fossils do tend to be very small. So if you've got small fossils, you can't just walk out and look for them on the ground. You have to get them out of the sediment in other ways. So at Great Fossil Site, the plant fossils are actually just collected with a bunch of sediment. So you don't just pick them up individually, they're collected with a bunch of sediment, and then they need to be separated from the sediment. So one of the processes that is used to do this is sieving. So up here in the upper left is a picture of some big tubs full of water, and people are there sieving the sediments and separating the organic remains and fossils from some of the other sediments, basically the dirt. Over here in the top center is a fan with one of these sieve trays, and it's blowing on the fossils to help air dry them. Once the fossils are air dried, volunteers go through and sort them. So normally when I go to the site, the material's already been sorted, at least to the extent that things that are thought to be plant material are separated from things that are thought to be animal material. So I don't see the animal stuff. Then once that amount of sorting is done, the fossils can be sorted further by sorting them by types of fossils and maybe trying to put preliminary identifications on them. So there are some pictures here. In the bottom left here is me at work with a paintbrush, just picking through the fossils. So again, these are very small. So you can't really manipulate them with your hands. Using paintbrushes or maybe forceps, basically like little tweezers you would use to pick up insects, are the kind of tools you'd use to move these fossils around. For storage, the fossils are actually stored in vials. So there's an example here at the upper right. This is a small vial that may contain 1 seed, or it may actually contain a bunch of seeds because they're very small. And over here on the right-hand side of the screen are just some examples of fossils. And I'll be showing a lot more examples later in the talk, but maybe a fruit, something that's unidentifiable with some holes in it, and at the bottom right here, what looks like a tendril. All right, so we've got the fossils removed from the sediment. We've got them sorted. How do we identify them? Well, the site doesn't come with a field guide. So it's not like you can go out with your field guide or your flora and try to key out what you're looking at. So that's where things kind of get a little bit more difficult. So one of the things that I'm trying to do as I research on the site is document different fossils and what I think their identifications might be, so that people at the site can actually help use this to build a field guide for staff and volunteers and students. So the pictures on the slide are sort of how I would go about identifying things. One thing that you can do is look at floras, fossil floras that are similar in age to Great Fossil Site and have similar preservation of plant materials. So there are a few in North America, Neogene floras from North America that have similar preservation. So on the left is showing some fossils from Brandywine and also from the Hattiesburg flora in Mississippi. A good place to look at floras that may have similar taxa in them, so genera and species, is actually Europe. Europe has a very good Neogene fossil record. There are a lot of fossils in brown coal deposits that are preserved in a similar way to the fossils at Great Fossil Site. So these have been documented extensively, from Germany and Poland, France, Italy. So they're great things to look at to get ideas for what might be in the Great Fossil Site flora. And I'll get into why Europe in particular in a little bit. Asian floras are also good to look at, and more material is being published from Asia as well as from South America. So at the bottom of the slide here are some fossils from Peru. And then also looking at modern plants. So I said this site was about 4.9 to 4.5 million years old, and that may seem really old, but from a paleontologist perspective, that was practically yesterday. So that means that most of the things that you will find at the site are probably, at least genera, are probably still living today. So you can look at the seeds of modern plants to try to get an idea of what's in the flora. One good resource for this is USDA. They actually have a fruit and seed atlas, as well as a lot of pictures online. You can also go to herbaria, so collections of plants, or I've even purchased seeds sometimes to get for comparison. So here's an example of just a comparison of a seed from another fossil flora and gray fossil site. So on the left is Xanthozylum tiffnyi. This is a type of seed that comes from a plant, the common name of which is prickly ash. It's actually in the same genus as Sichuan pepper. So this seed was documented from Europe. There are also similar seeds documented from the Brandon lignite flora. Oh, sorry about that red dot there. And on the right here is a seed from gray fossil site. So you notice that it looks quite similar with sort of this elliptical structure. This is the hilum, the micropyle at the top, and another opening, the rafal opening at the bottom. So can compare the features and see that they're quite similar. So I just wanted to go through a couple of taxa from gray fossil site in detail to go through the process of identification and putting maybe a preliminary identification versus a final identification on a fossil. So the first of these is Cavalignum praetschettii. This was a fossil I published with Carolyn Siegert. So this plate of fossils on the slide shows different examples of this structure. This fossil is actually something we interpreted to be an endocarp. An endocarp, and actually I sort of have this slide out of order, so I will move it up so you can see it. An endocarp is actually a structure in a type of fruit we call a drupe. So a drupe is a fleshy fruit, and it's got a pit or a stone at the center. So it's something like a peach or a plum or a cherry. And that stone or pit is actually a hard inner fruit wall that surrounds typically one seed. So endocarps are something we find in the fossil record because, of course, the fleshy part of the fruit is going to rot away and not be preserved. So paleobotanists are really interested in endocarps. So this Cavalignum fossil we decided was an endocarp. It's the inner wall of a drupe. One other thing that I should mention about gray fossil site is I'm not the first paleobotanist who has worked on this site. So there have been a few paleobotanists before me, and one nice thing that they did that's been helpful in working on it is come up with a little taxon list here. So it's not complete, but they did write down some of the taxa that they thought were preserved at the site. And one of these is something in Symplocaceae. And this identification, so Symplocaceae is the sweetleaf family. This identification almost certainly corresponds to these fossils. And it's not a bad identification, honestly. One of the things you may notice about these fossils, so these figures 1 through 8 are side views of the fossils. 8 through 11 show the tops of the fossils. And one thing you'll notice probably right away is there are 4 very prominent holes. This is really unusual. So the seeds would be sitting down inside the endocarp, inside of those holes. So it's very unusual to have these open holes at the top of an endocarp, but it is one of the characteristics of the fruits in the sweetleaf family. So what is sweetleaf? Symplocaceae. This family is a family, I think, of about 300 species of plants in 2 genera. Almost all of them are in a genus called Symplocos. The map up here in the upper right shows the distribution. So the areas in black are the modern distribution. They occur in the Americas and also in the Old World and Asia to Australasia. And the areas in red show fossil records. So there's a very good fossil record in Europe. This is a recurring theme for fruit and seed taxa, and scattered records everywhere else. So these plants produce drupes. One of the unusual things about the modern plants is that their endocarps have been very well studied. So on the bottom left here is an endocarp from a modern Asian taxon. And next to that are two fossils. So the fossil record is also very good and also well studied. The center one's from Europe, and the one on the right is from Vermont. You'll notice that all of these have the little holes at the top. So the top views show these open holes, which are characteristic of Symplocaceae endocarps. So if we compare Cavalignum with Symplocaceae in detail, however, we start to see some differences. So the Cavalignum endocarps, or Symplocaceae endocarps, often have a collar around the apex, so this sort of almost ring. These are not on the gray fossil site endocarps. The Symplocaceae or Symplocos endocarps have a central canal in them. The gray fossil site endocarps, if you look in the center of the ones where the picture is in the apical view, have two canals in the center. So that's a little bit different. And then if you look at the base of the endocarps in Symplocos or Symplocaceae, there's a pit, so a tiny hole. In the gray fossil site ones, there are actually little points at the base. So there are some subtle differences in the overall structure of these between the modern Symplocaceae and gray fossil site. There's also differences in the wall structure. So the gray fossil site material have these fibrous walls with kind of these long cells in them. Modern Symplocaceae don't have that. They have some variations, but not this fibrous wall. So in the end, after considering in detail all of these differences, Caroline and I decided that we wouldn't put it in Symplocaceae. So this appears to be the only maybe truly extinct thing so far identified in the flora, at least extinct, and we can't put it in a modern genus, and we don't even really know what flowering plant family it would be in. So just one more example of doing a structural analysis of a fossil. So this is another fossil. This one I published with Zach Quirk. Final paper just came out this year, but it's been online since 2020. So this is a group of seeds that represent this taxon. And there are a number of Hemophyllasi, so plants in the witch hazel family, that have been identified from gray fossil site. And one of them was the genus Coralopsis. So in this project, we were looking at these seeds and trying to evaluate, are they Hemophyllasi? Do they belong to Coralopsis? So what is Hemophyllasi? Well, Hemophyllasi is the witch hazel family. There are some members that are native to North America. Probably one of the more common ones is the witch hazel. There are several pictures here. It's commonly cultivated, but it also occurs, you can also find it in the woods. Also the witch alder down here in the lower right. So Hemophyllasi is distributed quite broadly around the world, North America, but also getting into North and South America, Africa, Asia, and it's divided into 4 subfamilies. One of these subfamilies is Hemimelidoideae, and this subfamily is very interesting because of the way it disperses its seeds. So plants in Hemimelidoideae have what we call ballistic seed dispersal, which is exactly what it sounds like. They shoot their seeds, and this is how they disperse them. So they have capsules in which the seeds occur. So up here in the center of the slide is a picture of the capsule. And as the capsule dries out, it actually puts pressure on the base of the seeds. And at a certain point, they will be shot out of the capsule. And this mechanism is sometimes compared to the mechanism of a rifle shooting a bullet. So there's a time-lapse series here on the left that actually shows fruit shooting one of these seeds. So this is interesting, but it's also a little frustrating, because what botanists think is that the seeds of different plants in this family tend to all look similar to one another because their shape is constrained by this ballistic seed dispersal mechanism. So they're all kind of small seeds, they're smooth. They tend to have a little bit of ornamentation, so a scar at one end that points towards the top of the seed. They may be a little tapered towards the bottom, but the differences are small, you know, a little bit of differences in size, maybe a little difference in shape, and some differences in the scars at the tip of the seed. So how do you as a paleobotanist determine whether you can put it in one of the modern genera of Hemophyllasi, or the witch hazel family? So in this case, what we did was we looked at the scars at the tip of the seed, in particular what shape they took. So in modern Coralopsis is a shrub, or it's a genus of shrubs and trees that occurs in Asia. There are probably around 30 species. So there's some pictures of the modern ones here on the right. In their seeds, one side of the seed shows a scar, and the other side shows a larger structure called a facet. So in looking at these seeds, most of which, unfortunately, we only had one face of the seed or the other, so we couldn't put it all together, it appears that some have smaller structures, which we called scars, and others have larger structures that we called facets. So at the end of the day, we put the gray fossil site seeds into Coralopsis. And I'll come back around to this later because this has some interest for us in terms of the biogeographical connections of the site. All right, so with that done and sort of going through two plants in detail, I now just want to go through a series of slides that show some of the diversity of plants at the site, just to illustrate what's there. So first up, there are some conifers in the fruit and seed flora. One of these is Taxodium. So Taxodium is the bald cypress. There are examples of 4 seeds on this slide. The seeds are probably about 1 centimeter in length. At the bottom left here is pollen, which also occurs at the site. And a student a number of years ago described some little branchlets and leaves. So there are multiple parts of this plant at the site. For comparison, on the right-hand side of the slide are some pictures of modern Taxodium or bald cypress. So modern seeds in the upper right, they're really irregular in shape. So in the flora, the seeds tend to look like maybe little bark chips or something. They're very odd. And then on the bottom is the tree. So these get to be very large trees. They are native to the southeastern US. Normally in their natural habitats, they're often growing in wetlands. These particular trees are cultivated. They're pretty widely cultivated. And of course, one of the interesting things about Taxodium is it's one of the few genera of deciduous conifers. So this one's actually in its fall colors and it'll drop its needles. Some other conifer material that I haven't fully identified are small cones, so probably seed cones, and what appears to be maybe a single cone scale. So of course, in a seed cone, the seeds lay on the cone scale. And I think this cone scale on the bottom right may belong to a pine or something similar to that. One of the things to note is there are 2 impressions on it. And of course, in pines, the seeds lay side by side on the cone scale, and they often leave an impression on that scale. All right, so angiosperms. For angiosperm material, a lot of things are either fruits, seeds, or endocarps, but there are other structures in the flora. So I'll show examples of a lot of different things. First up, these are some structures from angiosperm trees and shrubs. So I've already talked about Coralopsis. Here are some nice Coralopsis flowers next to it. I've tried to put some pictures of the modern plants next to each of these just to give an idea of what they look like. Upper right is hickory. So this was described in 2014 by Huang et al. And the modern hickory, of course, next to it. Bottom right is tupelo. This is Nyssa. I think it's also called gum. This is another tree native to the southeast. Blatternut. So these plants produce these inflated fruits, and seeds have been described from the site. And then a project I'm working on right now is the prickly ash. So this is Anthoxylum. Remember, this is the one related to the Sichuan pepper. So these are trees. It's called prickly ash. I don't have the picture here, but the trunks are often covered with these gigantic, they look like spines or thorns. They're prickles. So there are two species in the site, one with smooth seeds and one with ornamented seeds. This slide shows examples of structures from flowering plant, shrubs, and climbers. So the only shrub on the slide is actually rubus. So this is the raspberry or blackberry. There are rubus endocarps in the flora. So these are technically endocarps and not seeds. They're come from very tiny sized drupes. Passiflora, the passion flower, is something that I described recently from the site. This is a climber, and I'll talk a little bit more about this one later. It's interesting because this group has a very poor fossil record. Sargentodoxa, which is another climber. This has these very smooth seeds. This is a native of East Asia. On the bottom are Chinese moonseed. So this structure is actually an endocarp and not a seed. It comes from a member of the moonseed family, and you'll notice that it has this very characteristic ornamentation on it, sort of in a crescent or almost horseshoe shape. So this was described in 2010, and at the bottom left, grapes were also described. So on the left are fossil grapes, on the right are modern grapes. These are some examples of structures from herbaceous angiosperms, so plants that are not woody. So examples of tiny fruits of knotweeds. So this is kind of a weedy plant. A little black seeds that might belong to something like an amaranth. This is another weed. Member of the mint family. So these are nutlets. In the bottom right is possibly the spiny fruit of a cocklebur. So this is a member of the aster family. And then bottom center is fruits of a plant known as beggar tick. So this is something in the aster family as well. These, I'm a little bit concerned, might not be fossils. They could be contaminants. They're pretty rare in the flora. Finally, just a couple of other things that occur. One of the more interesting ones is cane or bamboo. So this is actually pretty unusual. You don't find things like this in the fossil record much, but it's basically a type of grass. You'll notice that the fossil has these striations on the stem and the very characteristic joint in the stem, which you can also see here in river cane, which is a bamboo that's native to North America. And then there are a few other structures. This little thorn in the center here might come from something like a raspberry. This little curly structure, maybe it could be part of a tendril. It's not entirely clear. All right, so this table just kind of gives a summary of the macrofossil flora. I'm not going to read it. The interesting thing about the flora, so it gives the taxon name, the structures, the common name, and also the modern distribution of the taxa, which is shown on the right. So if you look at the modern distributions of these plants, no obvious pattern might jump out at you, but there is a pattern here. And the biogeography of the site is really rather interesting. So for the last part of the talk, I want to talk a little bit about the biogeography. So Great Fossil Site really has what I would call a Laurasian or Northern Hemisphere biogeography. So it's made up of taxa whose distributions developed, between 66 million years ago to the present in a world that was very different than ours in some ways, much warmer with different geography. So what's going on at the site? Why do these things have these Laurasian distributions? And I just want to give, I guess, a little preface here. So this chart shows, it's basically an oxygen isotope chart, but it tracks temperature. So it basically shows the trajectory of Earth's global average temperature between the time that the dinosaurs went extinct, so that's on the right, about 66 million years ago, to about the present, which is on the left. So what you may notice is that things start out very warm, and the temperature starts to drop, it plateaus, it goes up a little bit, and then it starts to drop again. So gray fossil site is in one of these areas where it's dropping. But in the early part of the Cenozoic, it was very warm. In a gray fossil site, we have some plants that no longer occur there today. So, you know, how did they get to North America, or how did they get to where they are today? The biogeography of these plants was influenced, like I said, by changing temperature, but also by the changing configuration of continents. So that's shown in this series of globes on the right that goes from the present day back to, I guess, the late Cretaceous. But these are hard to see. To understand northern hemisphere plant disjunctions, what you really need to see is a polar view. What we're talking about is plants moving sort of in a circle around the pole. So in the early Eocene, or earlier in the Cenozoic, this map shows the early Eocene, which is about 56 to 48 million years ago. Plants can move in different ways than they can move today. So there were two routes of plant dispersal between North America and Eurasia. These were the North Atlantic land bridge, which was in the North Atlantic, and the Bering land bridge, which was in the North Pacific. So this globe is looking down on the North Pole. North America's here, Asia's over here, Europe's at the top. So North America on the right, Asia on the left, Europe at the top. Tennessee is actually shown by the letter T. You'll notice that with the North Atlantic land bridge, plants are moving through Greenland to Europe and North America, and the Bering land bridge from Western North America to Asia. In the Eocene, the Earth's temperature was also very warm. So there weren't big Arctic ice sheets blocking the distribution of plants, and more importantly, plants that today grow at relatively low latitudes could grow at much higher latitudes in the Eocene. So how do we know it was warm? Well, we have the global temperature curve, which is important, but we also have fossil evidence. And the fossil evidence is telling us it was warm, the types of plants and animals that were living there. So one very striking example is shown here. And I know this doesn't look that striking necessarily, but this is a piece of a vertebra from an animal that was related to crocodiles and alligators. And this was found in the Canadian Arctic archipelago. So it was very high latitude site where this crocodile-like animal was living. And it's thought that they weren't really very cold tolerant then, you know, even basically using modern animals as proxies. It's thought that, you know, they had similar tolerances in the past that they do today. So the Arctic would have been very warm if there were these crocodile-like animals living there. And this isn't the only thing. There are other things at these high latitudes. So I guess what I'm getting at here, the important thing is if you want to understand modern plant distributions and you want to understand gray fossil site, you really do have to understand this early Eocene world, because these plants at that time could expand their distributions across continents that aren't really, you know, as floristically connected today, at least for these plants that live in warm areas. So this is the modern world. The Bering land bridge would be in the North Pacific region, the North Atlantic land bridge in the North Atlantic region. So the arrows show approximately where these land bridges would have been. And the plants that were moving in the early Eocene in these land bridges are today tend to be restricted to very particular areas of the world. One of these areas happens to be southeastern North America or eastern North America. Also Western North America, part of Europe, and probably in terms of diversity, definitely most important, Eastern Asia. So why don't they occur everywhere today? Well, part of this is global cooling. So the poles have gotten a lot colder. And plants that can't tolerate that cold can't live at high latitudes today. So they can't make use, even if land masses are close to one another, they can't make use of high latitude connections. There are also other physical barriers that have developed. So for instance, the North Atlantic has been spreading. It's been widening over time, and it's put distance between Greenland and Europe. Also, we've had things like mountain building, and along with that, continental drying. So all of these things are barriers to plant movement, especially if these plants require warm, wet environments. So what are we seeing at Gray, and how does it fit into this? Well, we see what are some typical patterns of plants at Gray, or these typical Northern Hemisphere relic taxa occurring at Gray. So one of these is the hickory. So hickory has a very common plant distribution for the Northern Hemisphere today. It lives mainly in Eastern North America and Eastern Asia, and you can see it gets a little down into Mexico, but its fossil record is more widely distributed. So it's in Western North America, it's in Europe, it's in Japan, and it's up here, you know, in Northern Asia. So its range has contracted over time. And of course, it still does occur in Eastern North America today. But not all the plants that occur at Gray are still in Eastern North America. So we've talked about one of those. This is Coralopsis. So Coralopsis, the winter hazel, is a genus of about 30 species. It lives primarily in Asia. So its modern distribution is shown at the map in the lowest right-hand corner of the slide. So it's mainly in China, Japan, Korea, those sorts of areas. But if you look back in time, it's much more widely distributed. So there's evidence from seeds, which are shown with the red circles, from leaves, which are the green squares, and also wood, which is the blue hexagon on the top. So in the Paleogene, about 66 to 23 million years ago, it had a pretty wide distribution. In the Neogene, maybe a little bit less. And maybe that's because the Earth is getting colder, or maybe we just haven't found the proper fossil deposits. In the present day, it's restricted. So for some reason, it went extinct in Eastern North America. Why certain taxa went extinct in Eastern North America and survived in Eastern Asia is not always entirely clear. There are other examples of these East Asian genera that are found at Gray fossil sites, so 4.5 to 4.9 million years ago, and are no longer found in Eastern North America today. One of these is Sargentidoxa. This is a climber. It's actually found at 3 sites in Eastern North America. So it's found in 2 Miocene sites and at Gray. So Gray's the youngest occurrence, and it must have gone extinct at some point after that time. Another one of these is Cinnamonium, mood seed family. Again, this is an East Asian endemic that's gone extinct in Eastern North America. And Cinnamonium also has a broader fossil record. So the other interesting thing we're starting to find at Gray is some evidence of neotropical connections. So this is something that's just started to come out. One of the plants that I've worked on with this possible neotropical connection is Passiflora. So of course, the neotropics, we're talking about tropical America. Passiflora is a genus of more than 500 species. Its distribution is shown in the map in the upper right in the Americas and also Asia to Australasia, but really most of the diversity is in Northern South America, and it's less diverse everywhere else. So this taxon, this passion flower, occurs at Gray fossil site. And I guess the interesting connection is that it's one of the few things with this connection between North America and South America. Another example of a taxon with a potential neotropical connection is Xanthoxylum. So Xanthoxylum, the distribution of Xanthoxylum today is shown on the bottom in the Americas and also in the Old World. Again, although this map seems to show it occurring everywhere, it's really mostly in the tropics. So it appears to be another tropical connection in the flora. This is interesting because it's a rare connection. The question is, are these plants moving up from the south? Where are they coming from? In the case of Passiflora, it's hard to say. There are fossil records in Europe and South America, both. And the fossil record is so sparse, it's hard to make any secure connections. In the case of Xanthozylum, there's actually a great Northern Hemisphere fossil record. So it could be that this Xanthozylum was distributed, or this prickly ash was distributed quite widely in the Northern Hemisphere, and its current tropical distribution is a movement lower in latitude in both hemispheres, rather than maybe something coming from the south to the north to gray fossil site. And just one other little interesting side note about these tropical things, or these things that occur more in the tropics today, is that in both the case of Passiflora or passionflower and the prickly ash, the species, or at least in the case of prickly ash, one of the species at gray fossil site appears to be related to one of the more temperate modern forms. So in Passiflora or passionflowers, most of the taxa occur, or most of the species occur in Northern South America. There are very few that are native to North America, and there are only 2 that are widespread in Eastern North America. One of these is the maypop, or the purple passionflower, and its distribution is shown in this map on the right. So the gray fossil site taxon looks very similar to that one, and it's named Passiflora appalachiana now. So it appears to be in the range of the modern taxon. So maybe this more cold hardy or temperate lineage was already at gray by the Pliocene. In looking at Xanthozylum or prickly ash, the interesting thing is that the one of the gray species appears to be related to Xanthozylum americanum. And this is one of the American species, again, that's very temperate. And in the case of Xanthozylum americanum, its major distribution is actually north of Tennessee. So it's actually north of gray fossil site today, which I find very interesting. So I'm interested to see how this whole story develops with these potential tropical connections to the site. So to conclude, gray fossil site is an important flora for understanding the evolution of North American or Eastern North American vegetation. It gives us one of the few data points in the Neogene, and it tells us something about how the flora developed and how it was changing. So there are a lot of connections to other places in the Northern Hemisphere. There have been some extinctions since the Pliocene, and we also know this from other floras in Eastern North America. And interestingly enough, maybe we're starting to develop some southern connections or some neotropical connections. And so with that, I'll take any questions.
Dr. Hermsen, thank you so much. That was awesome.
Thank you.
Yeah, I learned so much. That was really great. And it's super important work you're doing. It's really refreshing to see someone who's studying plant fossils instead of animal fossils. So yeah, I think we've got some questions already going here, so I can start covering some of those.
All right.
So do you see these questions? I can read them out just so everyone else can hear them, but you should be able to see them too. But
yes.
The first question is from Kathleen, who asked, how and when was this site discovered?
Yeah, I meant to cover that, and I guess I forgot it. So it was discovered in the year 2000. It was discovered when a road construction project was occurring, and actually, I don't know if I can zoom back here. Not easily. I was going to try to show the site because you can see the road. There it is. All right, yeah, so this road, as far as I understand, was being built, and the site was found while the road was being built. And I was told that they actually sort of relocated the road a little bit to preserve the site, thankfully. So, so yeah, it wasn't found actually that long ago.
Okay. Yeah, I'm curious, like the first person who would have seen that, like how that, they're like, oh, hey, there looks like there's some bones here or something.
I think what happened, and
Yeah.
this is just from memory or what I kind of vaguely remember reading, was that someone may have hit something with construction equipment.
Yeah.
Which, probably isn't so uncommon either.
Yeah. Yeah. Pretty cool though. So we have a question from Kevin Burke who asks, have viable seeds been found in any fossils?
So not a great fossil site, and I don't think that anything would be viable from the site. I'm trying to remember what the oldest viable seeds have been. I know there have been seeds that are 2,000 years old, and I believe older that have been germinated. But I think something this old that probably would be an impossibility. And nothing that I've seen, I guess you could CT scan things that aren't broken, but nothing that I've seen suggests that any embryos or anything like that would be preserved. So it's pretty doubtful for something this old.
All right, we have a question from Simone Rosa, who asks, some of the fossils listed here are classified as non-native invasives by native plant societies. Should the definition of native plants in North America be adjusted, redefined to include these plants in the fossil record?
Right. So I guess I would say probably not. I mean, we're probably taking this from the perspective of what was the flora like, prior to European arrival and introduction of all these non-natives. So, I don't think going back in the fossil record 4 million years ago or more really, I guess, tells you that, well, I mean, it's not that it can't tell you anything about the ecology of North America today, but these plants aren't really integrated into the flora anymore. Mm-hmm. And especially when you're talking about these non-native invasives, they can really take over the landscape and cause a lot of damage. I mean, one thing I've really noticed around here is the Asian knotweed has just taken over large areas, and it's crowding out the native vegetation. So I guess the vegetation, I really think when you're talking about something that's 4.5 million years old, the vegetation today is probably not adapted or really co-evolved with it anymore.
Okay, we have a question from an anonymous attendee who asks, can you talk more about the material that you think might be contamination? What does this mean for the material, and how does that impact your findings?
That's a good question, and I'm not entirely sure how to answer that. I don't think so far, with how the site's been interpreted, that it impacts much. A lot of these things are widespread things, but it is something to be concerned about when you're dealing with a young site, when you're dealing with things with this type of preservation, and where the pit, you can see it in the background there. Of course, you're probably not seeing my Q&A screen. So the pit is pretty open a lot of the time. So it is something you have to bear in mind. I know when the material from Indiana was described, it was something they were concerned about as well. And I think they addressed it with some sort of carbon dating, and I don't really know how they went about that. But in terms of truly establishing if something's contamination or not, it might be worth looking into. I guess my suspicion with these things is they're rare. It is something that might grow around the area today, and they're really, really nice looking. So that all kind of makes me a little leery, but it doesn't mean they're not fossils. So I think if I were to publish them, it'd be something I'd have to think about further and figure out how well I can establish whether they're fossils or not. For other things like the cinnamonium or something, I don't really think it's a big issue. It's not something that people are going to be growing in the area or that's going to be growing on the site. Plus, a lot of these things are really found, I didn't really mention it, but because a lot of the planning and excavation is done by people who work on vertebrates, they've been very good about mapping the site and which squares things come from and exactly where they're found. So, you kind of know that things are coming from all around the site. I would feel more comfortable saying that's not contamination. It's probably something that's actually in the sediments, because how much stuff is going to be washing into all areas of the site as you're excavating it. So yeah, it's always, I think, something of concern when you're dealing with material of this type that's so nice and so well preserved and also relatively young.
Okay, we have a really interesting question here from Elena, who asks, have any Asemina or pawpaws or other Annoni fossils been found in this site or others in the Southeast? I imagine that would draw another interesting connection to South America.
Not that I know of.
Okay.
And you know, I guess going back to that South American connection, it's always interesting whether these are southern or northern connections. You know, like I mentioned with Passiflora and with Xanthozylum. With Xanthozylum, I think more study will show whether it's likely to come from the south or the north, but with Passiflora, who knows?
Okay, and folks, feel free to keep asking questions. I think we've answered all those for now. I actually have a question for you, though. I was curious, and you may have covered this, but what is the extent of the site that has already been excavated? I mean, how much? I mean, is there a good bit of area that still needs to be excavated? Do you think there's a possibility you're going to find even more species, or?
That I'm not entirely sure of.
Okay.
I don't know how much they still have to excavate. I think probably still a lot. You know, it's going along. But the thing is, it's not just the excavation, it's also the processing. So they've excavated a lot, but there are also tons of sediment that hasn't really been processed yet. So there's still a lot of stuff coming out. And the interesting thing about this from the perspective of a paleobotanist is because you're getting so much stuff out, you're having people look at it, it's more likely that you're going to find rare things, you know, things that are rarely preserved in the flora. So that's interesting too, because, you know, it gives you the chance to maybe find something that's only occurs occasionally in the fossil record.
Neat. So there's a good chance there'll probably be some more fun stuff in the future.
I hope so. Well, actually, I'm working on another, fingers crossed, fun project now, because I think there may be another Passiflora, so we'll see.
Oh, neat. Yeah.
Yeah,
that'd be cool. Yeah, I'm kind of curious what the Passiflora, I like maypops a lot. I'm curious what Appalachiana may have tasted like. Guess we'll never know. All right, so have you ever tried them?
I know.
Yeah, they're pretty good.
Maybe I've had a little of Edulus, but not the maypop. I used to see it when I was in Ohio, but we actually planted one. I live in New York, so obviously. So we planted one by our house, and surprisingly, it has continued to grow. Yeah.
Once you plant them, it's hard to get rid of them. So.
Well, we didn't know if it would survive with the winter, but
cool.
It's huge this year. So.
That's great. So I see another question popped up. So anonymous attendee asks, how often do you identify something to species level? And you might have mentioned, but what percentage of the species are you finding are still extant?
I don't think that anything has been assigned to a modern species.
Okay.
And I think this is, some of this may be more a question of philosophy and whether you should take something that's, you know, just a partially preserved thing and put it in a modern species or not. And, you know, if there's an age cutoff for that. So it doesn't necessarily mean that none of these were modern species per se, but a lot of times I think paleobotanists, especially as you get into older sediments, will be more likely to put it in a fossil species than put it to the modern.
All right, well, I think that about wraps it up for our questions. Thanks again for presenting today, Dr. Hermsen. That was really great presentation. And
thank you.
And if folks do have more questions, they can always send an email to me. I'll share that in a second. Oh, we got one more. Well, just a thanks so much. So.
Well, thank you for attending.
Yeah, and that was from Kathleen. Okay, so I'm going to go ahead and share my screen now. I just have a few last slides that I want to share about the upcoming event. Again, thanks everyone for attending tonight. Again, like I said, we're going to have a recording of this webinar that's available to the public, and there's a link right there for it. And you should be able, sometime early next week, it should be up on the website. I also want to talk a little bit about next week, or not next week, next month, October 21st at 6 p.m. Eastern Standard Time. We're going to have Ricardo Holdo, who is a professor of ecology at the University of Georgia, who's going to be presenting on some of his research investigating the factors that determine the structure and composition of tree communities in tropical savannas and grasslands. And as always, if anybody has any questions or suggestions, feel free to reach out to me. My email is isabo@atlantabg.org. And definitely, if you'd like, I encourage folks to check out our website to find out more about what we're doing here at the Conservation and Research Department. And that is atlantabg.org/conservation-research. And we also have an Instagram for any of you folks out there who are into the social media universe. And our Instagram account is @AtlantaBGConservation. All right, thanks a lot, everybody. Have a good evening.